Hi, this is Dr. Rosario, and this is our third video in our series introducing you to the world of microbiology. In this video, we're going to talk about our groups of microorganisms. So we're going to talk about the taxonomic hierarchy, the three domains of life, how we write the names of microorganisms, the different characteristics of our groups, and finally, the sizes of our microbes. So remember in our very first lecture, we talked about the diverse roles for microorganisms, right? They have so many different important jobs on this planet. They decompose our waste. They produce nitrogen and oxygen for us. They help to keep us healthy. They're important in farming, in pharmaceuticals. They are important in our food production. So microbes, they have many different roles, and they can also be found everywhere. They are ubiquitous. They can be found deep in the Earth's crust, down in polar ice caps. They can be found in the ocean. They can even be found inside of our own bodies, right? They can be found everywhere. Remember, microbes are organisms too small to see with our eyes. So even though we can't see them, they are there. They're everywhere. The science of classifying organisms is called taxonomy. Now, taxonomy really dates back very far in human history, even to the earliest times of Aristotle, the Greek philosopher. He had a system he called the ladder of life. Now, the ladder of life, he put humans at the very top, right? We were going to be the most important organisms. As we began to understand our world a little bit better, Carl Linnaeus came along and he developed a system of taxonomy that's generally still what we use today, classifying living things based on their physical appearances. So you can look at a sheep and a snake, right? And you can see that they are different from one another. They are in two different classifications. Sheep are mammals, snakes are reptiles, right? The physical differences that we see, sheep have fur, snakes don't. Sheep have legs, snakes don't, right? So we can start to differentiate living things just based on their physical characteristics. And this system was used for a very long time to group and organize living things. Now, one of the drawbacks to Carl Linnaeus' system was that it's based on those physical characteristics. When we look at microorganisms, particularly when we look at bacteria, a lot of them look the same, although they behave differently. So we had to come up with a different way to classify our microorganisms. Generally, we use biochemical characteristics to sort out which organisms belong with which group. So things like what kind of chemical reactions they perform, what kind of waste products do they make. And that has been a very useful tool for grouping and organizing living things. As we've progressed and we've learned more about the characteristics of living things and the molecular biology of living things, we've started to group living things not just based on their physical characteristics, but also on their evolutionary relatedness, right? So phylogeny is a taxonomic scheme in which we group living things not just based on the way that they look, but on how related they are to one another. And the way we determine how related organisms are is based on the similarity of DNA sequences. This system was developed by another Carl. This is Carl Woese, and he used what's called the 16S RNA sequence. So all living things have ribosomes, right? Ribosomes are the cell components that make proteins. And all living things have a piece of their ribosomes called the 16S RNA. By using the DNA sequence for that 16S RNA, we can compare very different organisms to one another because they all have that 16S RNA sequence. And using that system of comparing the DNA sequences, we can determine how related organisms are to one another. The more closely related they are to one another, the more similar their DNA sequences are. The more different that sequence is, the less related organisms are to one another. So we can look here at what we call the phylogenetic tree of life. And this scheme, what it does is it shows you the three main groups of living things. So we have three domains. We have bacteria, archaea, and eukarya. And then within each of those, we have groups of organisms. Now, as you look at this tree, it's based on evolution. So down at the bottom is what we call the common ancestor, right? The earliest common ancestor of all living things. All living things are thought to have evolved from early life, right? And we have a ancestor that we all share. And then as time progressed, evolution drove differentiation, right? Different organisms evolved over time. So each place where you see a branch is where there is a common ancestor between the two parts of the branching point. And the closer the branching points are together, the more related the organisms are to one another. So I want you to notice that bacteria are off to one side by themselves, and archaea and eukarya are grouped together on another branch. It turns out that while we used to group bacteria and archaea together because they're both prokaryotic, right? They don't have nuclei in their cells. Archaea are actually more related to eukaryotes than they are to bacteria when we look at their gene sequences. Okay, so we have divided all living things into three main groups, right? Our domains, bacteria, archaea, and eukarya. And then within each one of those domains, we can break them down further into smaller groups. So each domain is made up of kingdoms, and then within each kingdom, you have different phyla. Each phylum is made up of different classes. Classes have orders within them. Within orders, you have families. Within families, you have genuses. And then within genuses, you have species. Right? This is what we call the taxonomic hierarchy, the way that we group and categorize living things. Now, the way that I remember this taxonomic hierarchy is I have a mnemonic, right? Deer, king, Philip, came, over, for, ginger, snaps. That's a little bit easier for me to remember than domain, kingdom, phylum, class, order, family, genus, species. Right? And there's other mnemonics out there. You want to find the one that works the best for you. This just happens to be the one that I know and that I learned. So we're going to really focus primarily on the genus and species level categories because this is where we get our names for our organisms. So naming organisms is based on the genus and the species. So here we can look, this bacteria in this picture is Escherichia coli. That's a bacteria that you've probably heard of, right? E. coli. Escherichia coli gets its name from the genus Escherichia and the species coli. Now, the reason they're called Escherichia and coli is really kind of arbitrary. Escherichia, that name came from the scientist who discovered it, right? His name was Theodore Escherich, and coli comes from the location where this organism can be found. It's found in the colon of mammals, right? So Escherichia coli, that's where that name comes from. Escherichia, again, is the genus. Coli is the species. We can abbreviate this name by just saying E. coli, right? Now, we usually, when we're writing this out, we will always first refer to the full name with the genus and the species, and then after that point, we can refer to it with the abbreviation. Here's our second scientific name example for you. This is Staphylococcus epidermidis. Again, genus is Staphylococcus, species is Epidermidis. This one gets its name because staph means grape-like, and coccus means round. So Staphylococcus are these grape-like round clusters of bacteria. And then epidermidis refers to where we find this organism. It is found on the skin. So again, the genus is Staphylococcus. The species is Epidermidis. There's lots of different staphylococci. For example, you may have heard of Staph aureus, right? Staph aureus is also sometimes called MRSA because it's a method. There are strains of Staph aureus that are antibiotic resistant that can occur in the hospital environment. Okay, so that scientific name has two main components. You have the genus and you have the species. So let's take a look at that again. And I'm going to use E. coli as my example. So you have Escherichia and coli. When I write this out, Escherichia is going to be capitalized because that's the genus, and coli is going to be lowercase. And then I want to make sure that I underline the scientific name if I'm writing it by hand. Right, so the proper way to write out a scientific name is to include the genus and the species, genus capitalized, species lowercase, and then if you're handwriting it, you're going to underline it. If you're going to type it out, you just want to italicize it instead. So you're going to see Escherichia coli in italics. It is a pet peeve of mine when I'm reading popular news articles and I don't see the scientific names written correctly. Now, throughout the semester, we're going to have a variety of writing assignments, discussions, and other things. When you write scientific names, make sure that you're writing them correctly. Okay, so we've talked about the way that we classify living things, right? We have our three domains, bacteria, archaea, and eukarya. And remember, your bacteria and your archaea, they are one group of cells, right? They are your prokaryotic cells, those that don't have nuclei. And your eukarya are those cells that have nuclei, right? Eukarya literally translates into true nucleus. So as we're going through this semester, you kind of want to be able to group these living things based on their shared characteristics so you can differentiate between them. And we covered that a little bit in our first lecture. So we're not going to cover archaea much this semester because we don't know of any that cause human disease. We're also not going to talk much about algae this semester. So your 5 main groups of organisms that we are going to cover are bacteria, fungi, protozoans, helminths, and viruses. And then within viruses, we also include prions. Viruses and prions are not composed of cells, so they're kind of their own group by themselves. So it's useful to build yourself a table so that you can look at those main characteristics of living things. So remember, I covered all of this in the first lecture, but let's just go over it real quickly. Bacteria, they're prokaryotes, where our fungi, our protozoans, and our helminths are all eukaryotes. Viruses and prions, they don't have cells, so they have no cell type. As far as the number of cells, bacteria are single-celled organisms, right? They only have one single cell. Fungi can be single-celled or multicellular, right? Your yeasts are single-celled, but your molds and your yeasts are single-celled, but your molds and your mushrooms are multicellular. Protozoans are all single-celled eukaryotes. Helminths, which remember those are worms, those are all multicellular. And again, viruses and prions, they're neither, right? They don't have cells. When we look at the cell walls of our organisms, bacteria have a cell wall composed of peptidoglycan. Fungi have a cell wall composed of chitin. Although we really don't talk about plants in this class, the third type of cell wall is your cellulose cell wall. Protozoans, they don't usually have a cell wall. Sometimes they have something called a pellicle, which is a protein coating on the outside. Helminths, which are animals, don't have cell walls. And of course, again, viruses and prions have none of these things because they are not cells. The last thing that I want to look at is our size of microorganisms. Remember, microorganisms are so small that we cannot see them with our eyes. Now, we're in a science class, so we use the metric system. We measure size in meters rather than feet and yards. A meter is a little bit larger than a yard, so it's bigger than 3 feet. We are going to be talking about organisms that are on the order of micrometers in size. So 1 micrometer is 1/1,000,000th of a meter. So your average prokaryotic cell is about 1 to 2 micrometers in size, and your average eukaryotic cell is usually somewhere between 10 to 50 micrometers in size. So you can see we have this relative scale here for you to help you kind of visualize the different types of cells and organisms that we're going to study this semester. So prions are infectious proteins. They're not even alive, right? They're going to be the smallest of our pathogens that we're going to study because they're just proteins. They're biological macromolecules. From prions, the next largest of our microorganisms, and I use that term very loosely because they're not alive, is our viruses. Viruses are usually on the range of nanometers in size. So the polio virus is about 30 nanometers in size. It's a very small virus. There are some other viruses that can even be up to a micron or even 10 microns in size. But most viruses are somewhere between 30 and 1,000 nanometers in size, right? So they're very, very small. Next up will be our bacterial cells. Bacterial cells, again, there's a lot of diversity here, but your average bacterial cell is 1 to 2 micrometers in size. And then the next group of microorganisms up is our unicellular eukaryotes. So unicellular eukaryotes, remember that includes our yeast, our protozoa, and even some of our algae. Those cells are approximately 10 microns in size, sometimes even larger. Finally, our largest group of microorganisms is going to be our multicellular eukaryotes. Our multicellular eukaryotes range in size, but we are multicellular eukaryotes, right? So we're quite large. Your tapeworms, which I mentioned in the first lecture, those are multicellular eukaryotes. So they can be quite large, but they can also be very, very small and have to be seen with a microscope. So there's a lot of diversity with those. It includes our multicellular fungi, our molds and our mushrooms, and our helminths, our worms. Okay, so in this lecture, we really covered our different types and groups of our microorganisms and how they are related to one another and how they compare to one another. As we move on, we're going to start looking at some of the chemistry of living things.